Let's explore the antigen-antibody reactions used in serology testing. Precipitation occurs when a soluble antigen combines with its corresponding antibody, provided they are present in the right proportions. This leads to lattice formation, where the antigen binds to the FAB sites of two antibodies. The result is the appearance of a visible precipitate. Examples of precipitation tests include double diffusion, also known as the Ouchterlony technique, single diffusion or radial immunodiffusion, immunoelectrophoresis, and immunofixation. Agglutination works on a similar principle but involves particulate antigens rather than soluble ones. When antibodies bind to these particulate antigens, lattice formation occurs, bridging the antigens and creating visible clumps. Examples include direct agglutination, which is widely used in blood bank testing, passive hemagglutination, where red blood cells are treated with protein antigens, and passive latex agglutination, where antigens are attached to latex particles. Agglutination inhibition is a two-step process. In the first step, patient serum containing antigen is mixed with a limited amount of antibody reagent. In the second step, an indicator is added, such as the same antigen bound to red blood cells or latex particles. If the patient's serum contains the antigen, the antibodies bind in the first step and are no longer available to react in the second step. This produces a positive result, meaning no agglutination occurs. On the other hand, if the patient does not have the antigen, the antibodies remain free and react with the indicator, producing agglutination. This is considered a negative result. Common examples include the hemagglutination inhibition test for rubella and latex agglutination inhibition tests for other viral infections. Our next principle is complement fixation. In this method, antibody and antigen are allowed to combine in the presence of complement. Next, we add an indicator, which is sheep red blood cells coated with hemolysin. If complement was already fixed in the first step, it will not be available for the indicator system, and no hemolysis occurs. This is a positive test. On the other hand, if complement was not fixed, it reacts with the indicator, causing hemolysis, which represents a negative test. Complement fixation has some limitations. The serum must be heat inactivated, stored serum often becomes anti-complementary, strict quality control is needed, and it primarily detects IgM antibodies. Next, we discuss radial immunodiffusion, also called single immunodiffusion. In this technique, antibody is incorporated into agar plates, and serum samples are placed into wells. As antigen diffuses, it forms a circular ring of precipitate. We measure the ring's diameter. The Fahey method is kinetic, read between 6 and 12 hours, and follows a logarithmic relationship. The Mancini method is an endpoint method, read at 24 to 48 hours, and shows a linear relationship. Now let's look at double diffusion, or Ouchterlony's method. Here, an antibody is placed in a center well of agar, while patient samples and standards are placed in surrounding wells. As diffusion occurs, visible precipitation bands form. The position of the bands depends on antigen concentration and rate of diffusion. This method is often used to identify antibodies associated with autoimmune disorders. Next up, immunoelectrophoresis, or IEP. This technique combines gel diffusion with electrophoresis. Serum proteins are first separated by electrophoresis, then a trough is filled with antibody. Antigen and antibody diffuse toward one another, forming arcs of precipitation in the equivalence zone. The size of the arc reflects antigen concentration, while abnormal patterns may indicate monoclonal gammopathies. Clinically, serum IEP is used to detect monoclonal gammopathies, while urine IEP is used to identify Bence-Jones proteins or free light chains. This image shows serum protein electrophoresis, also known as SPE. It is a laboratory method used to separate and analyze the proteins found in serum. The results are displayed as peaks or bands, with each peak representing a different protein fraction. At the top blue, we see the normal pattern of serum protein electrophoresis. The tallest peak on the far left is albumin, which normally makes up the largest fraction of serum proteins. Moving to the right, we see progressively smaller peaks representing the globulins. First, the alpha-1 region, followed by the alpha-2 region, then the beta region, and finally the gamma region on the far right. In a healthy pattern, albumin is the tallest peak, while the other regions are smaller and relatively balanced. Now, below this, bottom, red, we see the pattern for monoclonal gammopathy. Here, the albumin peak is still present on the far left, along with the smaller alpha-1, alpha-2, and beta fractions. However, what stands out is a very tall, sharp spike in the gamma region. This spike represents the overproduction of a single type of antibody, also called a monoclonal protein or M protein. This finding is characteristic of diseases such as multiple myeloma or Waldenström's macroglobulinemia. In summary, the normal pattern shows a smooth distribution of proteins across albumin, alpha, beta, and gamma regions. In contrast, monoclonal gammopathy shows a sharp, narrow spike in the gamma region, which is a hallmark of abnormal plasma cell proliferation. Let's review immunofixation electrophoresis, also called IFE. Immunofixation is a laboratory method that combines protein electrophoresis with immunoprecipitation. It is highly sensitive and relatively easy to interpret. Here's the procedure. First, the specimen is applied to 6 positions on an agarose plate. Next, electrophoresis is performed to separate the proteins. After separation, monospecific antisera are applied to 5 lanes, leaving the 6th lane as a reference. If an antigen is present, a band of antigen-antibody complexes will form and precipitate. Finally, the plate is washed and stained so that the bands can be visualized. The great value of immunofixation is in its clinical application. It is particularly useful for classifying monoclonal gammopathies, helping to determine which heavy and light chains are involved. This makes it a key tool in diagnosing conditions like multiple myeloma and related plasma cell disorders. This diagram represents immunofixation electrophoresis, a powerful tool used to identify monoclonal proteins in serum. On the far left, you see the serum protein electrophoresis reference lane. It contains multiple broad bands, representing the normal distribution of serum proteins such as albumin, alpha-1, alpha-2, beta, and gamma globulins. Next, the individual lanes correspond to IgG, IgA, IgM, kappa, and lambda light chains. In this case, a distinct sharp band appears in the IgG lane, and another sharp band appears in the lambda light chain lane. The absence of similar bands in IgA, IgM, or kappa lanes confirms that this is a monoclonal IgG lambda protein. This result would be reported as IgG lambda monoclonal gammopathy, which is often associated with disorders such as multiple myeloma or related plasma cell dyscrasias. In summary, reference lane, SPE, normal distribution of proteins. IgG lane, distinct monoclonal band. Lambda lane, matching monoclonal band. Final interpretation, IgG lambda monoclonal protein. Let's walk through the different immunoassay techniques. Rocket immunoelectrophoresis. This technique is similar to radial immunodiffusion, but it uses electrophoresis to make the reaction faster. The gel contains antibody, and the patient's serum, which carries the antigen, is placed into small wells. Standards and controls are also added. When an electric current is applied, the antigen moves through the gel quickly. A cone-shaped precipitate forms that looks like a rocket. The height of the rocket is measured and compared to a standard curve. Countercurrent immunoelectrophoresis. In this method, 2 columns of wells are cut into the gel. Antigen is placed in one column and antibody in the other. When an electric current is applied, the antigen and antibody move toward each other. If they match, they form a visible line of precipitation. Radioimmunoassay, or RIA. This method is very sensitive and specific. It can detect either antigen or antibody. It works on competitive binding, where patient antigen competes with labeled antigen for antibody binding. After washing away what is not bound, Radioactivity is measured. The lower the radioactivity, the higher the concentration of patient antigen. Examples include testing for hepatitis antigens and antibodies, total IgE with RIST, and allergen-specific IgE with RAST. Enzyme immunoassay, also called EIA or ELISA. This method uses a sandwich technique. First, antibody is fixed onto a solid surface, such as a bead or a microtiter well. The patient's sample is added, and if antigen is present, it binds to the antibody. Next, an enzyme-labeled antibody is added, forming a sandwich with the antigen in the middle. When substrate is added, the enzyme causes a color change, and the intensity of the color corresponds to the antigen concentration. Proper washing between steps is critical for accuracy. Examples include HIV testing, pregnancy testing for HCG, hepatitis testing, and detection of viral or bacterial antibodies, enzyme multiplied immunoassay technique, or EMIT. This technique is especially useful for measuring small molecules like drugs and hormones. In a positive test, the drug in the patient's serum binds to the antibody, leaving the enzyme free to act on the substrate, producing color. In a negative test, the antibody binds to the enzyme portion of the conjugate, blocking its active site, so no color is produced. Nephelometry. In nephelometry, a patient's serum substance reacts with specific antisera to form insoluble complexes. Light is then passed through the suspension, and the scattered or reflected light is measured. The amount of scattered light is directly proportional to the number of insoluble complexes present, and results are compared to standards. Nephelometry is commonly used to measure complement component concentrations and antibody levels, including immunoglobulin G, immunoglobulin M, and immunoglobulin A. Immunofluorescence. There are two main types of immunofluorescence techniques. In the direct method, fluorescent-labeled antibodies are added directly to patient tissue on a slide. After washing, the slide is examined under a fluorescent microscope. In the indirect method, or IIF, patient serum is added to reagent tissue containing a known antigen. After washing, a fluorescein-labeled antiglobulin is added, and the slide is examined under a fluorescent microscope. Examples include testing for anti-nuclear antibodies, also called ANA, and the fluorescent treponimal antibody absorption test, abbreviated FTAABS, fluorescence polarization immunoassay, or FPIA. In this method, reagent antibodies and fluorescent-tagged antigens are added to patient serum. In a positive test, patient antigen binds to the reagent antibody, leaving most of the tagged antigen unbound. These unbound tagged antigens rotate quickly, reducing the amount of polarized light produced. In a negative test, when no antigen is present in patient serum, the tagged antigen binds to the reagent antibody. These antigen-antibody complexes rotate more slowly, giving off an increased amount of polarized light. This technique provides a sensitive and reliable way to measure antigen-antibody reactions in clinical immunology. Let's talk about serial dilutions and how they relate to evaluating test methodology in the clinical lab. Serial dilutions. When testing for infectious diseases, we often compare two specimens, one collected during the acute stage and another during the convalescent stage, usually about 2 weeks later. To confirm infection, there must be at least a 4-fold rise in titer, or what is also called a 2-tube rise. This significant increase indicates that the patient's immune system has mounted a response to the infection. Sensitivity. Next, let's discuss sensitivity. Analytical sensitivity refers to the ability of a test to detect very small amounts of a substance. Clinical sensitivity, on the other hand, measures the test's ability to correctly identify patients who actually have the disease. A test with 100% clinical sensitivity will produce no false negatives. Specificity. Now, specificity is just as important. Analytical specificity is the ability of a test to detect the correct substance without interference from cross-reacting substances. Clinical specificity measures how well a test avoids false positives by giving a negative result when the patient does not have the disease. A highly specific test ensures accuracy and reliability in clinical decision-making. Let's dwell further on sensitivity and specificity. Think of sensitivity as the test's ability to correctly identify people who actually have the disease. The formula is true positives divided by the sum of true positives and false negatives, multiplied by 100. When false negatives increase, sensitivity goes down. Now, specificity tells us how well the test identifies people who do not have the disease. The formula is true negatives divided by the sum of true negatives and false positives, multiplied by 100. When false positives increase, specificity decreases. So remember, N is for negative. Sensitivity deals with false negatives. P is for positive. Specificity deals with false positives. When we evaluate test sensitivities, we divide methods into non-lattice and lattice techniques. Non-lattice methods are more sensitive. These include radial immunoassay, abbreviated as RIA, enzyme immunoassay or EIA, fluorescent immunoassay or FIA, and nephelometry. These methods can detect substances at very low levels, close to 0.001 milligrams per milliliter. Lattice methods are less sensitive. These include countercurrent immunoelectrophoresis, or CIE, complement fixation, abbreviated as CF, agglutination techniques, flocculation, also called precipitation, rocket electrophoresis, radial immunodiffusion, or RID, Ouchterlony or double immunodiffusion, immunofixation, abbreviated as IFE, and immunoelectrophoresis, abbreviated as IEP. These tests generally detect substances in much higher ranges, close to 500 milligrams per milliliter. So remember, non-lattice methods are more sensitive, lattice methods are less sensitive.